So one of the best things that I do is that I co-direct a group at the Canadian Institute for Advanced Research. It has a new name, and it's called Child and Brain Development, and I do that with Professor Tom Boyce, who's sitting in the front row. And one of the things that we're interested in is how our experience gets embedded in our biology. And a group of us have met with autism researchers and clinicians to talk about the themes that have come out of the last 10 years of our research program that addresses these issues, and which one of them might be relevant for questions around autism. So one of the things we can ask in this room is, why are we all different? And many, many years ago, you might have heard people say, well, it's all nurture. We're born as a blank slate, and the environment writes on it. Whereas another group might have said, maybe a biologist like me, that nature is really important. It's all in our genes. And now we know that this nature-nurture dichotomy is dead. In fact, we can't even take how much is nature in the genes and add it to how much is nurture in the environment and add those together and say, well, that's why we have these individual differences. In fact, there's an interaction between the genetics and our environment. And these genes that predispose us to behave in certain ways or to give us certain risks for certain disorders, they come in different forms. And some of them buffer our experience or our brain development, and some of us make our brain development and the behavioral outcome from that behavioral development be more susceptible. And so we can see that we have different genetic variants. Many of them have been identified. There's lots of them. And some of us protect us, whereas others make us more susceptible. And within a typical normal population, Tom Boyce and others have said that some of our children, when they're born, are like orchids, whereas others are like dandelions. And so the dandelion kid that is not sensitive to context particularly could be transplanted from place to place. It doesn't matter too much what happens to that child in terms of its early experience. It's always going to do pretty well. Whereas the orchid child needs, like probably my two kids, need to be watered and kept in the right humidity, and then they flourish. And so far mine are flourishing. Whereas if that orchid child is born into a neglect or born into misfortune, then they do really poorly. And so there are genes that predispose all of us to be buffered from certain environments, to really do exceptionally well given what we experience. And these are some of the things that we study. In fact, our genes are listening to the environment. The DNA is static in that it's there in all our cells and it's all the same, but it's listening in the sense that it responds by the genes. The genes make proteins, they make chemicals, and the amount of that protein or where it puts that protein, is it going to put it in the liver, in the neuron, in another cell, has to do with the environment that, for instance, developing brain experiences in utero and also once the child is born. And we know now that there's this new area called epigenetics, and it's been all over the paper. You might have heard about it. And this is a mechanism for nurture. It's a way that we now understand how our experience gets embedded in our biology. And so if you take identical twins, their DNA is the same, and yet you probably all know identical twins, and as they grow up, you see quite a few differences. It could be differences in temperament, in the way that they look. And if we then go in and look at their DNA, we find that DNA is wrapped around these proteins called chromatin, and when it's wrapped around, that DNA does not end up being particularly expressed, but when the DNA opens into a more open conformation, then it does get expressed. And so our environment acts like a dimmer switch to change the expression of our genes, the amount of enzyme in our brain, for instance, in a certain place, and it goes up and down, and that depends on our early experience and also our experience as we go through adolescence. So we can think about these gene-environment interactions in that way, and we've been studying the mechanism. And the next 5 years, we're trying to understand how these molecules are regulated in the developing brain, how the body is talking to the brain, and how we arrive at individual differences according to the susceptibility genes and this epigenetic phenomenon. And then finally, the other big area in our group has to do with critical or sensitive periods in development. And this is pioneered by Takao Hench, who was at our meeting from Harvard University. And you may know that our brains are like sponges, right? We always say, oh, my 2-year-old's brain is like a sponge. And really, our brains have windows of opportunity that open and close. And during development, if you're pregnant and you're, let's say, 6 months pregnant, around that time, if you play that baby music, my son would say, play the baby jazz. Some of you might like Bach, and others might like rock. And that once that baby comes out, it can distinguish between the type of music that it's heard. Or if you play from Janet Worker's work in British Columbia, if you play different sounds from your language, like da in English or da, which I can't say in Hindi, but it sounds different, when that baby emerges, it will have had that exposure about language, and it will know the difference between those sounds. And then that window of opportunity closes, and by the time you get like me and you try and learn another language, for sure you're going to have an accent. So there's windows of opportunities for listening, for seeing, for language, and also for social behavior. And so later on, children learn how to regulate their social behavior, how to have social cognition, how to read social situations. And we know that this window that opens and closes is regulated by molecules that Takao and others have discovered, and he and others are able to open that window, close it, make it longer, and make it shorter. And he and others think that these windows in brain development and the information coming in may have something to do with how that brain is wired differently or not. And so that's another very interesting area of relevance. So I'm just going to finish up now. So when one thinks about normal or typical variation in all these different traits that have to do with autism, if one thinks about that, we all know people who are more shy socially or check their door before they leave several times. I mean, with me, it's always, where's my key? And some of us are better communicating verbally as compared to in writing. And I think if we graph the abilities of the people in this room, looking at many of these different characteristics that we are associating with autism, at least in a quantitative way, we would see that there's no one control. There's nothing that is, okay, we're going to compare it to that. In fact, we have this huge distribution. And then the same is true for the autistic kids, right? It's the same kind of huge variation from one end to the other in the spectrum. And then we're trying to figure out, well, what is the control? Who should we be comparing with whom in our experiments? And so I think one of the things we've realized in the last few days is we need to pay particular attention to the way we phenotype, that is, how we quantify these traits, not just quality in that you have it or you don't, but how do we measure those things? And I lost my train of thought. You're not supposed to do that when you're talking in public. But anyways, so you're right. And so that should be informative. And not only that, but we don't only want to quantify where those individuals sit in the typical and atypical population, but we know things change in development. You're on a developmental trajectory, and so there's a developmental way of thinking that something happens at this time, that's going to affect what happens later. And that's true with behavior and brain development, and all those time periods interact so that in the end you have a grown-up human being. And so this way of thinking, which doesn't particularly focus on autism or schizophrenia or Alzheimer's, how the typical brain develops and the requirement for that development to optimize health and promote optimal well-being for every child, which is one of the focuses of our group, those findings and the things that we're discussing, I think, have been really relevant and exciting, and it's been great to interact with the clinicians and the researchers.